Good morning, good afternoon, and good evening, everyone from all over the world, and welcome to our webinar titled Telemetry 101. This webinar has been sponsored by ADInstruments, so a big thank you to them for helping to make this event possible. Joining us today, we're very fortunate to have Dr. Phil Griffiths, a training and support specialist at ADInstruments in Europe. His presentation will highlight a number of common applications of the ADInstruments small animal telemetry systems and showcase some exciting publications from existing users. I'm Sarah McFarland from the events team here at Inside Scientific, and I'm very pleased to be your host for today's event. I'm very pleased to welcome Dr. Phil Griffiths to the floor. Phil, thank you so much for joining us today, and the floor is yours whenever you're ready.
Great, thank you very much. Okay, hi everybody. Thank you for joining us today. So I'm going to talk about the new small animal telemetry system from ADInstruments today. So just to give you a little bit of information about me, I've got a BSc and PhD in neuroscience from the University of Bristol in the UK, where I've also had 4.5 years postdoc experience using a telemetry system to record arterial blood pressure from rats. In 2019, I joined Carha Sciences to work as a telemetry application specialist in Europe before I moved in February of this year to work for ADI, and I'm currently as a training and technical support specialist in the European team. Just to give an overview of what we're going to talk about today, we'll first look at why you might use telemetry and some of the alternatives to telemetry that are available, perhaps the traditional techniques that you might think of. We'll look at some of the specific details about the telemetry system from ADInstruments and the advantages of it as well, and how it integrates with the existing ADInstruments data acquisition system, the PowerLab and LabChart. The telemetry system can be broken down into a rat system and a mouse system, so we'll look at those independently of each other, and then finally we'll work through some research applications and showcase some exciting publications from customers. So to give you some background, the telemetry system I'm talking about today is the Carha Sciences telemetry system. And this isn't new technology. It's been around for about 15 years and was first sold under the brand of Telemetry Research before it was purchased by Millar in 2014. Millar carried out a lot of product improvements and introduced a few new products and features and things as well before the brand was changed again to Carha Sciences in 2017. ADInstruments purchased Carha in February 2021, so it's a really exciting matchup now between the telemetry technology of Carha Sciences and the existing data acquisition technology from ADInstruments. So Carha Sciences is now an ADInstruments brand. And with the Carha Sciences telemetry systems, you can measure a range of physiological parameters, whether they're cardiovascular parameters, cardiovascular pressures, or other physiological pressures, sympathetic nerve activity. There's a unique tissue oxygen telemeter as well. And as I've said, the systems are broken down into a rat system and a mouse system. At the moment, the mouse system is biopotential only, so both the rat and the mouse systems offer biopotential recording, and the rat system offers the pressure recording. So just to kick off the presentation a bit more, what are some of the alternatives to recording with telemetry? These can be broken down into acute recordings, the first of which I would consider the invasive recordings in anesthetized animals. These are one-off measurements, so you carry out your experiments maybe over the course of a day, and then at the end of the experiment, the animal's culled, and that's the end of the experiment. With these experiments, you can collect high-frequency data with high fidelity sampling, And your recording time is usually sort of a few minutes to perhaps a few hours, and it's continuous waveform data. So these kind of studies, acute blood pressure recordings maybe, or something like a PV loop study. And it's with the Millar catheters, or some sort of in vivo anesthetized neuroscience recording from the brain. These studies are really valuable, but you have to bear in mind the effect of the anesthesia on your physiological data that you're collecting. And as I've said, you're only collecting data from the animal over the course of one day. And then you have to repeat the experiment in another animal. So you'll find that you have to have quite a large sample of animals before you're getting statistically significant data often. Another example of acute measurements are non-invasive measurements, like non-invasive blood pressure using tail cloth plethysmography is a good example of this. And these are usually one-off or periodically repeated experiments. And they are quite time-consuming. It involves quite a lot of training of the animals, particularly with the NIBP to sit within this tube. And the recording time is usually limited to a few minutes, and it's non-continuous data. So rather than a waveform data that you'd collect, otherwise you're maybe getting 1 or 2 blood pressure values out for the recording time. So these are quite valuable for screening large numbers of animals or a colony of animals to make sure that it's either a colony where you might want them to be expressing hypertension, or checking whether hypertension is developing in that colony over time. But it's quite difficult to collect sort of publishable data with these systems, I think, because of the amount of training and time that's involved in using these systems. The final example that's not telemetry that I'm going to give today is the example of tethering. So this is where you might, these are chronic experiments where you might implant your electrode or your blood pressure catheter in the animal and then externalize it at the shoulder blades often, and then connect it via a tether to an external transducer or amplifier. A lot of evidence over time has shown that these studies restrict the movement of the animals during recording, leading to altered behavior and perhaps changes in your physiological activity of the animal as well. And they can be quite prone to infection where you externalize the electrode leads or the catheter through the skin. They're also prone to movement artifacts and noise, where the tether is a big aerial, basically picking up environmental electrical noise, and is also moving around and can influence the data. Recording time with this is perhaps up to a few hours a day and can be repeated over a few days or weeks if you're lucky that your signal is maintained. But a researcher has to be present throughout all of those recordings and watching that animal and during all of that time. So again, they're quite time consuming. What telemetry offers you is the ability to perform conscious, unrestrained data collection, physiological data collection from your research animals. Telemeters are fully implantable, so they're less prone to infection. And after a short surgical procedure, the animals recover to behave to behave normally and unencumbered by the telemetry device. There's minimal day-to-day maintenance. There's no need for researchers to sit there watching the animals while they're recording. Maybe say for going in once or twice a day to make sure the system is recording as you would expect it to be. And the data is coming through like you would expect. And you can have multiple animals running simultaneously. So when I was using the system as a researcher, I would implant rats in groups of 4, so that I would have up to 8 animals running simultaneously. So a group of 4 in a control group and a group of 4 in an experimental group. So you can get quite a high throughput going for your experiments as well. With telemetry, you can run long-term recordings over weeks and perhaps months. And this helps you to capture long-term variation in physiological parameters for aging studies, perhaps, or to look at disease progression over time. And although telemetry systems can come with quite a significant cost at the beginning, over time, they do actually allow you to reduce your costs because of the quality of data that you can collect with telemetry systems and the possibility of doing within animal controls. You can have fewer animals required per group to give you higher statistical power. And you can also reduce the staff maintenance. As I've said, you can free up staff time to perform other activities like data analysis or other experimental work while the animals are recording. Okay, so to move on and talk more specifically about the cardiovascular telemetry system from ADInstruments, as I've said, it's broken down into a rat telemetry system and a mouse telemetry system. The rat telemetry system comprises the TR181 smart pads, which are the data receivers for the TRM5 and TR5 series rat telemeters. The mouse telemetry system comprises the MT110T bases, which are the data receivers for the MT10B single biopotential mouse telemeters. The smart pads and the T bases also act as wireless power devices to provide power to the telemeters. And I'll explain what this means and what this means for data collection in the next slide. Common to both systems is the TR190 configurator system, which is the small box that you can see over to the left-hand side of both diagrams. And this system allows full software control of the telemetry hardware to reduce the impact of handling and stress on your animals. So you don't need to handle the animals in order to set up the system, and is actually a really nice system and makes the telemetry system really easy to set up and use. Then you also need your Power Lab and Lab Chart for data acquisition, data capture, and data analysis. So I've mentioned that the smart pad and the T-base generate wireless power. So what does that actually mean for the telemetry system? Well, they use electricity to generate a magnetic wireless power field, which passes through the cage base and the skin of the animal and is picked up by the telemeter and converted back into electricity to power the telemeter electronics. So what this means is that while you're recording from your animals in the home cage, there's actually continuous power available to the telemeters. And this removes the experimental limitation of battery life. So compared to other telemetry systems where you're constantly having to manage the battery life and how much longer you've got left of recording with that particular telemeter, with the Caja Sciences telemetry system, you don't need to worry about that. During home cage recording, the telemeters are powered continuously, and this opens up the possibility of really continuous recording, firstly, and being able to capture lots of spontaneous physiological activity, but also long-term recording for monitoring physiological parameters over a really long period of time. It also removes the need for refurbishing telemeters. So the rat telemeters can actually be explanted from the animal and reused, and the number of reuses depends on the application and the type of telemeter that we're talking about. And the mouse telemeters are actually designed to be used once and then disposed of, and priced for that as well. So the configurator system, as I've mentioned, allows full software control of the telemetry hardware. So the telemetry system uses 40 independent transmission frequencies. So each telemeter and smart pad or T-base is paired on its own transmission frequency to avoid crosstalk between telemeters and interference between animals. So you can be confident that the data transmitted from a telemeter is picked up by the specific smart pad or T-base that you want it to be. So this is all controlled using the configurator system. And it's really easy to set up and use, and there are videos on the ADInstruments website that show this as well. The configurator system allows you also to carry out monitoring and diagnostics of the system to maintain optimal recording conditions and make sure that everything's working as it should be. It's worth pointing out that this is not a data acquisition system. It's not required for data acquisition. It's solely for the control and the configuration of the telemetry system. Data acquisition is provided by the PowerLab data acquisition hardware and LabChart software from ADInstruments. And there's really seamless integration with this data acquisition system. The SmartPad and T-Base analog outputs connect to the analog inputs of the PowerLab, and you can perform real-time data capture and analysis with LabChart 8 and LabChart Lightning. So you can see in the video that this rat is implanted with a dual pressure telemeter measuring arterial blood pressure and left ventricular pressure. And that waveform data is being collected in real time. And we've also got an analysis channel going on here, calculating the PDT from the left ventricular pressure waveform. LabChart Lightning is ADInstruments' newest data acquisition and analysis software, which was released last year. And this is really powerful for the telemetry system moving forwards, including unlimited data analysis or data acquisition and analysis channels, custom calculations, cross-recording analysis, and really easy data management as well. So with the amount of data that you can record with the telemetry system, it can be really easily managed in LabChart Lightning. LabChart 8 Pro at the moment has the built-in modules for analysis of waveform data. So that's the blood pressure module, the ECG analysis module, and the heart rate variability module are just 3 examples of the modules that will be useful for analyzing your telemetry data, particularly for cardiovascular customers. And these soon will be introduced into LabChart Lightning as well. This is a good opportunity to introduce the PowerLab C as well, which is a new data acquisition device from ADInstruments, and it provides simple, reliable, low latency connection between digital devices. So it can be combined with this instrument interface, which is sitting on top of the PowerLab C in this image, to take the outputs from the SmartPads and the T-Base and take them through into LabChart. And this is really the future of ADInstruments data acquisition. So watch this space for more information about the PowerLab C in the near future. So just to summarize, up to this stage of the webinar, that the Caja Sciences telemetry from ADInstruments uses wireless power technology to remove the experimental limitation of battery life, to allow continuous recording from your telemetry devices, but also long-term recording as well over a long period of time. The telemeters, both the rat and the mouse telemetry system, benefit from a really high sampling rate at 2 kHz for improved data accuracy. The configurator system allows full software control of the telemetry hardware to reduce any impacts of handling of your animals. And there's really easy integration with the existing ADInstruments data acquisition system, the PowerLab and the LabChart. So I'm going to move on now to talk more specifically about the rat telemetry system. So the rat telemeters and the rat telemetry system offers the ability to measure pressure, physiological pressure, and that's achieved by the incorporation of the Millar Mikro-Tip pressure sensors. And I'll talk a bit more about what that means for the quality and accuracy of the data, the pressure data that you collect in the next slide. The rat telemetry system also offers co-housing mode, so that's a really important feature for improving the welfare of your animals, but also, and another side to it, is allowing you to customize your experiments to collect the maximum amount of data from each animal that you use as well. And I'll talk a bit more about that in the upcoming slides. all of the rat telemeters have a backup battery, which helps to facilitate continuous recording in the home cage, but also allows sampling up to 5 meters away from the smart pad for between 4 and 6 hours, depending on which telemeter it is that you're using. And this is ideal for behavioral studies. So it really opens out the possibility of carrying out more complicated experiments, not just with the animals sitting in their home cage, but taking the animals away from the home cage as well. And also all rat telemeters accurately measure core temperature and can report core temperature as well, alongside the parameters advertised with each telemeter model. So I mentioned that the rat pressure telemeters incorporate the Millar Mikro-Tip pressure sensor, which is a solid state pressure sensor, 2 French diameter, which is 0.6 millimeters, located at the catheter tip. And this technology offers unrivaled long-term accuracy, fidelity, and reproducibility compared with gel or fluid-filled catheters. And the reason for this is that pressure is actually recorded directly at the source where this sensor is implanted. So this is at the end of the catheter. If you're recording arterial blood pressure, for instance, this part of the catheter is implanted in the artery, and you're recording the signal directly via this small window here is how the pressure waveform is detected. With a gel or fluid-filled catheter, you're relying on the transmission of that pressure waveform along the catheter to a transducer that's located in the telemeter body. And that can introduce effects of gravity, animal movement, and obviously you run the risk of that catheter bending or a blood clot forming in that catheter, causing your signals to disappear. And all of that's removed with this solid state technology from Millar, meaning that you can record really accurate and pressure waveforms with the Caha Sciences rat pressure telemeters over a really long period of time. You get an excellent frequency response because you're recording directly at source. There's no need for the signal to be transmitted through the gel or fluid as well, so meaning that you get an excellent frequency response, which is great for measuring dPdt accurately from your left ventricular pressure waveform. And this also supports unique applications, particularly intracranial pressure, ICP, which I'll talk in a bit more detail later. But this is a really low pressure waveform and can be really accurately captured with the rat pressure telemeters. There's been a lot of talk and concern from ethics committees and bodies in the past about the need to singly house animals for telemetry experiments. And this is overcome in the Caha Sciences rat telemetry system through the co-housing mode, where you can implant two rats with telemeters and house them in the same cage and record from both telemeters simultaneously. This really improves the welfare of the animals because rats, after all, are a social species, so they like to be together. And we've got a number of customers that have reported changes in the physiological data that they observe compared to singly housed animals when rats are housed together. Co-housing mode also offers the possibility of customizing your experiments as well. So using the same feature, 2 telemeters can actually be implanted in a large rat over 350 grams to mix and match between any of the rat telemeters available at the moment and measure up to 4 parameters. So that gives you the option of really maximizing the amount of data that you can collect from each animal as well. This video just shows an example of co-housing mode in action. One of the rats is implanted with a blood pressure and biopotential telemeter recording arterial blood pressure and ECG, and the other rat is implanted just with a single pressure telemeter. And you can see that these guys are housed together and recording in real time into LabChart. At the moment, there are 7 rat telemeters available. There's the single pressure telemeter, dual pressure telemeter, a pressure biopotential telemeter, pressure sympathetic nerve activity telemeter, single and dual biopotential telemeters, and a tissue oxygen telemeter. And we'll look in a little bit more detail about some applications of these telemeters later on. So just to introduce the mouse telemetry system now. Traditional mouse telemetry is limited by the size of the implant and therefore the size of the battery that can be included in the implant as well. A small battery means that you're limited in the amount of power delivered to the telemeter electronics, meaning that recording time is restricted, and also the sampling rate has to be kept relatively low as well as the cost of refurbishment and the time that takes up too. The Caja Sciences solution is a battery-free telemeter which receives continuous wireless power from the T-base to allow recording 24 hours a day, 7 days a week at 2 kilohertz sampling rate. So this is great, again, for your long-term recordings and your detection of spontaneous events as well with continuous recording. At the moment, the mouse telemetry system just offers a biopotential telemeter. And this is designed for subcutaneous implantation in mice over 22 grams. It's designed and priced for single use. So you implant it in the animal, it's really easy to use, and then you dispose of it at the end of your experiment. The T-base is redesigned compared to the SmartPad to deliver optimized wireless powering to the telemeter to ensure that the telemeter receives wireless power as much as possible, and this allows you to record a biopotential, so either ECG, EMG, or EEG at 2 kilohertz. Alongside the biopotential, the T-base also allows you to measure an index of activity as the mouse moves around the cage through the wireless power field, and also to detect whether data has been received by the T-base as well. So you can see here, this is an example of a mouse recording ECG as it moves around the cage. And the real benefit of being able to record continuously means that, as I've said, you can track spontaneous activity. And an example of this here is an unfortunate event where a mouse implanted with an ECG telemeter suffered a myocardial infarction as a result of the experiment that it was undergoing. But what you can see is that you were able to track the development of the MI over the course of an entire day. So in the morning, the ECG waveform was pretty normal. There was some development a bit later on in the day in the evening, and then over a 2-hour period, the whole event was captured right up until the point of death. With a traditional mouse telemetry system, you wouldn't have been able to capture this data because you would have been restricted in the amount of recording that you could do. So you might have only taken an hour's recording sometime in the middle of the day, where you would have seen this fairly normal ECG recording, and then the following morning you'd be presented with a dead mouse and you didn't know what happened. Whereas with this system, you're able to track it, capture all of this data, and use that as part of your study for reporting that data as well. In terms of long-term recording, you get really good signal integrity as well. So this is an example of 2 ECG waveforms recorded from different mice 60 days after implantation with no filtering to show that the signal integrity is really good and data quality is still there after that time. And it's also possible to record longer than 60 days. This is an example, but certainly up to 90 days is possible, if not even longer. So the last part of the webinar, I'm going to talk through some applications of the telemetry system, both the rat and the mouse system, and also highlight some publications from customers, which are pretty nice. So starting with cardiovascular physiology, the telemetry systems can be applied in studies of hypertension using the rat blood pressure telemeter, single pressure telemeter, or the pressure biopotential telemeter to record arterial blood pressure and ECG. In studies of cardiac function and MI, you can use the rat pressure biopotential telemeter to record left ventricular pressure and ECG, or the dual pressure telemeter to measure left ventricular pressure and arterial blood pressure. Then you've also got the biopotential telemeters, so the rat single biopotential telemeter and the mouse biopotential telemeter to record ECG too. And then finally, from a cardiovascular physiology, this is a bit of a whistle-stop tour, but you can also carry out studies of cardiovascular control using the 56SP arterial pressure and sympathetic nerve activity telemeter to record arterial blood pressure from the descending aorta and renal sympathetic nerve activity as well. So this is looking at influences of the autonomic nervous system on blood pressure. And you can see in this example, this rat's implanted with a 56SP telemeter recording arterial blood pressure and a really nice, clean sympathetic nerve activity waveform, which is possible thanks to the 2 kHz sampling rate of the telemeters. And using LabChart as well, this is an integrated signal of that raw waveform there as well, showing that you can carry out real-time data analysis too. And a great example of this telemeter in use was published in Nature at the start of this year by Jordan Square from the EPFL in Geneva in Switzerland and his collaborators at the University of Calgary in Canada. And they were using the 56SP telemeter to measure arterial blood pressure and sympathetic nerve activity to monitor the hemodynamics of spinal cord injured rats. And then looking at some treatment protocols using some stimulation as well. And this is a really, really nice paper. Even besides the telemetry, I'd really recommend that you go away and read it because it's a really interesting study. But it really highlights the power of the telemetry system too. So they were recording over a really long period of time. So they were recording up to 42 days after their spinal cord injury. And they were recording continuous data as well. So they were recording 24 hours a day, 7 days a week. And the quality and stability of the data that they were able to capture with the telemeter allowed them to confidently perform automated analysis as well. So a certain proportion of their data analysis was carried out without human intervention. So they were recording into LabChart, exporting the data into another data analysis software package and running this automated analysis. And they were confident to do that because of the quality of data that they were collecting with the telemetry system. They were also able to record under a number of situations in their experimental protocol as well. So they were carrying out continuous recordings where they were just monitoring in the home cage. They were able to carry out and monitor hemodynamic parameters during the spinal cord injury surgery as well. And then finally, they made some weekly assessments in this negative pressure chamber as well. So recording away from the home cage for a period of time to look at the effects of this negative pressure chamber on hemodynamics and how the rats were responding to after the spinal cord injury. So this is a really excellent example of the kind of data that you can collect with the telemetry system. So moving away from cardiovascular pressures into talk more about the intracranial pressure that I mentioned earlier. So because the solid state pressure sensors are so accurate and are able to record at really low pressure and very sensitive to small changes in pressure as well. This makes them very suitable for recording intracranial pressure in rats. The telemeter body is implanted in the abdominal cavity, as you can see here, and then using a 25 centimeter catheter, it's tunneled subcutaneously and can be implanted underneath the skull, either subdurally or epidurally. And a great example of this in action is from Sajeda Eftekari and her colleagues at the Danish Headache Center in Copenhagen. So they were able to record they were able to implant the pressure sensor epidurally and record a really, really nice, really low pressure. You can see in this example, it's below 5 millimeters of mercury, really nice waveform that they were able to analyze and extract other components of it as well in this top part of the figure here. Because what the sensors also offer is really long-term stability as well. So as you can see in the middle diagram, this is raw data or a mean from a daily mean from 10 animals plotted over 50 days. And they were able to get pretty stable recordings from all of their telemeters, with, save for maybe 1 or 2 days in a couple of animals where the signal was a bit higher for some reason or lower. But they were able to get really stable recordings for 50 days from their animals. And that's exemplified by this, how tight these error bars are down here in this final figure. And what's nice about this study is that they were able to carry out the experiment in different phases. So they had the recovery phase where they were looking at how ICP changed immediately post-surgery, and they show that it takes about 9 to 10 days for ICP to stabilize after surgery. They then had a physiological phase where they were just interested to see how ICP or the ICP recording in the home cage without any experimental intervention, and they were carried this out for a good 3 weeks. And then the final part of their experiment was carrying out some experimental interventions as well. So this is a really nice study and shows the quality of data that you can collect even at such low pressures and so stable over a long period of time as well. So some further applications of intracranial pressure would be in studies of stroke, traumatic brain injury, migraine and intracranial hypertension, which is what the guys in Copenhagen are interested in, and hydrocephalus as well. So to move on and talk about the some more neuroscience specific applications of the telemetry system, and this is using the rat single and dual bipotential telemeters and the mouse bipotential telemeter as well. The continuous and long-term recording is really a nice feature available for neuroscience studies. And the fact that you can sample at such a high sampling rate means that the frequency bands that you would expect to see recorded in EEG or even from deeper electrodes in the brain are really able to be captured by the telemetry system. Unlike with a tethered system or a head-mounted system, you see fewer movement artifacts because all of the telemeter is internalized. There's nothing to bang against the side of the cage, so you get really nice signal quality. And it's ideal for detection of spontaneous events like seizures during epilepsy studies and sleep as well. And the rat telemeters, as I've mentioned, have the 5-meter transmission range to the smart pad, so you can take your rats away from the home cage and carry out studies and experiments in mazes or fear conditioning as well. And a couple of examples of the biopotential telemeters in action for neuroscience studies come from these epilepsy papers. One from Professor John Jeffries from the University of Oxford in the UK, where he was using a dual biopotential telemeter to detect seizure activity via an EEG waveform, and also ECG to look at the effects of heart rate during and after the seizure. And you can see that he was able to record for 50 days from the telemeters and capture a really large number of seizure events, each of which they were able to analyze and look at the effects on heart rate during those seizures too. The other figure on this slide relates to this paper from these researchers at the University of Utah in the US. And again, they were using the single biopotential telemeter to record EEG activity and monitor seizure activity in rats. But they were able to record for even longer. So they had something like 18 weeks recording from their telemeters. And they were able to implant the telemeters right at the beginning, record some baseline inducing epilepsy. Then they induced epilepsy in their animals, carried out a dosing regime, and then split their animals out into 3 experimental groups as well for the final part of the experiment. So able to collect really lots of data from each animal and for really good statistical power. I mentioned the tissue oxygen telemeter, and I haven't really got enough time to go into much detail about this today, but apart from to say that it's a unique means of monitoring chronic tissue oxygen from the kidney and the brain, where it's been used so far. It's been used by researchers to look at circadian variability in kidney tissue oxygenation in this cell metabolism paper down here, and also looking at relating it to neuronal activity and looking at local ischemia in the brain as well. And then finally, I just want to highlight the application of co-housing mode by implanting 2 telemeters in 1 rat. So these guys at the University of Auckland in New Zealand, in Fiona McBride's group, implant the dual pressure telemeter and the 57Y tissue oxygen telemeter in a rat model of stroke. And this allows them to record arterial blood pressure, intracranial pressure, and brain tissue oxygen in their stroke model. So this is a really highly customized experiment specific to what they want to do, and using the co-housing mode to implant these telemeters in 1 rat, large rat, is allowing them to collect as much data as they possibly can to extract as much information from the rats and what's going on during their stroke model. So you can make unique combinations of physiological parameters, and as I say, to really maximize the use of each animal and to get some really interesting data. And again, these are really interesting papers to go and read about the blood pressure after stroke in rats, and some therapeutic approaches as well. And really, again, showcasing the use of the telemetry system. So just to sum up, the wireless power system of the Car Sciences telemetry system removes the experimental limitation of battery life and supports really long-term and continuous recording. The 40 independent transmission frequencies mean that its systems are really easy to use, and there's no interference between animals. And you're sampling with a 2 kilohertz sampling rate, which is really high for improved temporal resolution. The rat telemetry system incorporates the Millar Mikro-Tip pressure sensor for improved accuracy and expanded applications for pressure telemetry. And the co-housing mode improves the welfare of your animals, but also allows you to refine your experiments and extract as much data from your animals as you can. The mouse system is a battery-free telemeter, allowing that continuous and long-term data collection, which you can't get with other mouse telemetry systems, at a really high sampling rate as well. And finally, it's single use, which makes it very easy to use. So thank you very much for listening and attending. If you've got questions, I'll answer some questions shortly, but feel free to direct them to me at p.griffiths@ADInstruments.com. And specific sales questions can go to the different sales email addresses in the different regions. And for more information, there's loads more information available on the ADI website as well. Thank you.
All right. Thanks so much for that presentation, Phil. I am just going to flip to our Q&A session. So the first question here for you, Phil, is does the wireless charging of the implants cause the animals to heat up at all?
Yeah, that's a good question, common question as well. The short answer is no. And we've got some data that we collected a few years ago to support that as well. But basically, the wireless power technology is quite smart, and the field generated by the smart pad and the T-base is optimized based on feedback from the telemeter. So the telemeters constantly measure the temperature within the animal to make sure that they don't overheat. And if for any reason they are warming up, then they are able to disable the wireless power field as well. So there's a lot of built-in kind of fail-safes to make sure that doesn't happen.
Okay, great. And then a somewhat related question here. Someone has asked, does the telemetry systems, do they measure temperature?
So the rat telemetry system does measure temperature, yes. The mouse system doesn't. The rat system works that it measures temperature on a 2-hourly cycle. So you get an updated temperature measurement of the rat every 2 hours.
Okay, great. Another great question here. This question is, when reusing the EEG/biopotential units between different animals, can you address how or whether one can lengthen the lead wires if needed?
The answer to that is that you can't at the moment. So each time that you explant them, you'll have to trim the lead wire a little bit. And the best advice that I can give to maximize use is to keep the amount that you have to trim those leads to a minimum. So for EEG recording, you have to embed them in dental cement to hold them in place. So be quite sparing in using that dental cement so that you trim as little as possible. The leads are quite long as well, so they're, I think off the top of my head, they're about 25 centimeters. So you should get a good few uses out of them before, as long as you're careful as well, before having to buy a new one.
Great. All right, another really awesome question here. So this person has said, I work in respiratory neuroscience, and I was curious if there were any applications regarding plethysmography methods. Currently, we use a pit tag telemeter for temperature monitoring. Are there any applications for this telemetry regarding respiratory research?
That's a good question. In terms of plethysmography, no, there's nothing that I don't think that you can do with these telemeters for that. In terms of respiratory research, you could measure diaphragm or intercostal EMG to look at respiratory activity, and you can also measure intrapleural pressure as well using the pressure telemeters. But yeah, they're the two main applications that I can think of for respiratory research.
Okay, great. Another question here. In order to measure left ventricular pressure, where do you need to secure the transmitter?
So the body of the telemeter would be implanted in the abdominal cavity, and then the catheter is implanted in the left ventricle via the apex of the heart. So you go in through the diaphragm. There are webinar. There's a my colleague or ex-colleague Sarah Jane presented a webinar on the surgery approaches and in recording left ventricular pressure, which is available on the ADI website. And there are surgical videos on the ADI website as well, showing the surgical approach for implanting for LVP.
Great. We've got a couple more questions here. This is just a quick reminder to send questions that you have for Phil, and we'll try to get to them in the next couple minutes. So this question here is, can blood pressure and cerebral spinal fluid at the lumbar level be recorded simultaneously?
Sorry, so they want to record blood pressure and
cerebrospinal fluid, probably pressure as well at the lumbar level.
Okay, I'm not aware that anybody's measured CSF pressure in the spinal cord. It's possible. It just depends on the size of the sensor relative to the kind of space that you can put it in without causing too much damage to the spinal cord, I think. So because it's a 2 French sensor, which is 0.6 millimeters, beneath the skull, there's plenty of room for that to be positioned. In the spinal cord, around the spinal cord, it's certainly possible, I think, but it would just be a bit of a tight squeeze, I think.
Yeah, for sure. Our next question here is there co-housing possibility for the mouse system?
Unfortunately not at the moment. The mouse system, the mouse system can't work with co-housing. So the only option for that is to have an unimplanted cage mate in there with the implanted mouse. Yeah.
Okay. Great. And then this next question is kind of related. Is there a plan to scale up the mouse system to record multiple biopotentials and/or the core temperature?
I don't know the answer to that at the moment, unfortunately. It's certainly a question that people have asked of me previously about recording a couple of biopotentials from the mouse system. So at the moment, I don't think there are plans to do that, but something might happen in the future, perhaps.
All right. And in the interest of time, I'm going to make this the last question. This question is there anyone you could recommend with experience placing sensors in the rat pulmonary artery? I'm assuming pressure.
Yeah, I guess that's for pressure. I don't know anybody off the top of my head that does that. But if the person wants to contact me, then we can discuss it in a bit more detail, I think, rather than addressing that now. I don't think we've quite got enough time to talk about that now.
Yeah, for sure. Just a reminder, if you do have questions for Phil, his email is right here up on the screen. And those links are clickable, so that will open an email right to Phil or to the ADI sales teams. So thank you so much, Phil, for your presentation and your insights during the Q&A. It was really a pleasure to have you with us today.
Thank you very much. Thanks for having me.
All right, and again, if you do have any other questions for Phil, you can submit them now using that Ask a Question panel, or you can send them to him directly. And I also want to thank the audience for being here today. Thanks for taking time out of your busy schedules. I know everyone's on a lot of webinars these days, so it's really great to have you with us. As I mentioned at the beginning, the slides and a recording of today's webinar will all be available soon. So look out for an email giving you access to that recording and these resources in the near future. Before you go, again, we'd love to get your feedback on that survey. Let us know what webinar topics you'd like to see in the future. And finally, if you do have any other questions, last time, submit them through that Ask a Question panel. In closing, thank you again for taking part in this Inside Scientific webinar sponsored by ADInstruments, and we look forward to having you with us again soon.